Hello, and welcome to Genetics Chapter 2, or Unit 2, um, review video. So let's start right off with DNA mutation and repair. So, mutations, this is an important note, probably will be on the test. Mutations are the source of all new genetic information. So, there are no new alleles, no new phenotypes without mutations. Mutations are the only way we can get a different phenotype. Like, say, if we somehow develop the ability to have purple hair, then that would be from a mutation. So then mutations could be negative or positive. A lot of times we think of mutations as purely negative or purely positive, like, oh, mutations cause cancer. And they can cause cancer, but in the case of, say, it cause people to be born with purple hair, that might actually be pretty cool. That might be a positive thing. Or in the case of um, something more practical, when during before the Industrial Revolution, they were white. There was a type of moth that was white because there wasn't a lot of smog in the air, so they blended in with the birch trees. But then after the Industrial Revolution, yeah, they started a mutation caused some of them to have black wings, and that mutation took over. And that actually helped the moths because then there was so much smoke in the air that they actually blended into the smog a lot better. So that's how a mutation can be for the positive. Okay. 98% of DNA is non-coding. So honestly, most mutations do not change anything. Like if there's a mutation in a telomere, which is usually just like a bunch of repeats, and this turns into a U, or no, not a U, it would be like a T instead of an A, um, that doesn't really matter because it's telomere, like it doesn't get transcribed. Somatic mutations. These affect non-reproductive cells and do not pass to the next generation. This is what you would think when you have cancer. Um, so those affect stuff like your skin cells, your organs, basically anything that is not in reproductive cells, which would be insect cells, and that would lead past to the next generation, and that's where you get your genetic diseases, Huntington disease, etc. Gene mutations. So these affect a single gene and then therefore only a few proteins. These can still have big effects, like a single gene can be the difference between someone who's blind and someone who's seeing, but it's usually more localized. Chromosomal mutation is um, affects a lot of genes, and usually the people who have chromosomal mutations have a wide range of various issues, health issues or developmental delays. Something like trisomy 21 or Down syndrome is kind of um, an example of this, or Prader-Willi syndrome, which affects chromosome 15. But a lot of them are actually fatal, but the ones that are survivable, they usually have a lot of problems. So, types of gene mutation we have substitution, and that is one base converts to another. It's pretty simple. However, we have a bunch of other types of substitution. We have transition, which is purine converts to a purine. So A to G, or pyrimidine converts to a pyrimidine. So T to C, or vice versa. Basically, transition ones are like goes to like. There's transversion ones, and that's purine to pyrimidine, or pyrimidine to purine. So that's like to unlike. So transition, like to like, transversion, um, the two are the same. Uh, there's also indels, which is short for insertion or deletion, and that leads to a frame shift because if you say you have A, C, G, T, A, just random strands, um, if we have this regularly, then we would read it like this. This protein, this protein, this protein, or not protein, amino acid, this amino acid, this amino acid, this amino acid. But then if we delete this, all of a sudden it becomes AGT, AGT, GA. And then all of a sudden we make this amino acid, and then this would be, I don't know, whatever comes next. So that can be a huge problem because that basically destroys the entire protein the gene is trying to make. So indels are usually a lot more serious than substitution mutations. Um, and when an indel happens, it leads to a frame shift mutation unless you somehow delete 3 in a row perfectly. Um, Spanning trinucleotide repeats. So these basically, there are a lot of trinucleotide repeats, like a common one is CAG, and you have a ton of those in a row. But if you have too many of them, it can cause some problems, like Huntington's disease. And they can also affect gene expression. Um, so yeah, Huntington's disease is um, one that affects like the central nervous system, and basically these are normal. If you have a ton of these, it's bad. um Phenotypic outcome of gene mutation. So we have, this is how did the mutation affect the phenotype, what we see on the outside, because again, sometimes gene mutations do nothing, and sometimes they do a lot of damage or a lot of positive change. um So we have a missense mutation. This is a new codon encodes a different amino acid, leading to a change in the sequence in the protein. This usually is pretty damaging because, again, a different amino acid in the protein means that the protein will have a different structure, which means it will have a different function, or it will lose that function. So missense mutations are pretty serious. We have a silent mutation where a new codon encodes the same amino acid, meaning the same protein is produced. These are harmless. This is why we have so much redundancy in the translation code. It's to help build in a little bit of this buffer, which is in the silent mutations. We also have a nonsense mutation, and the new codon creates a stop codon, and it stops translation prematurely. This is pretty damaging because the protein's not fully formed, and then it can't fold into its structure, which affects function. So yeah, pretty bad. So missense and nonsense are pretty bad. Silent is fine. There's also the idea of loss of function versus gain of function. So, some mutations can give you more muscle. That's a gain of function. Mutations, you lose muscle. That's a loss of function. That's kind of the general idea. It's pretty self-explanatory. We also have conditional versus lethal. So, some mutations will only show up in certain places, or they won't affect you too much, or it's survivable. Um, Lethal is pretty self-explanatory. It kills you. So yeah. There's also forward and reverse mutations. So, forward causes a change in the phenotype, and reverse assumes that you already have you're not in the wild type, you're in a mutated version of the gene. So if the wild type is brown, um, if the wild type is brown, and you have the mutated that turns your hair blue for some reason, then the reverse mutation would change this blue back into brown. But it really only works if you already have it mutated. Like you can't really turn brown back into brown. The reverse can either affect the same gene. So that would be um probably that, so it can either like, if the change was say from an ATG to an ACG, and this somehow radically affected the protein so it turned blue, um we could have a reverse mutation that turned it back to ATG, and then it would go brown again. Or we could have a mutation on a completely other protein that overshadowed this blue protein. So say it used to be A, and now it's ACA, and this suddenly makes a brown protein that overshadows this blue protein, and then it also goes back to brown, but this is still mutated. So that's the two different ways reverse mutations can work. We also have For interactions between genes, we have intergenic and intragenic. Intergenic is interactions between two non-allelic genes. So that means that we have gene 1 and gene 2, and these code for different proteins, but they're interacting together. So like this example here, this one produces the brown protein that covers up the mutated blue protein. That would be an intergenic relationship. Intragenic means interactions between two alleles of the same gene. So we have one gene and then we have two alleles in here. This one says white hair, this one says blue hair. And they interact and maybe they create light blue hair. That would be an intragenic interaction. Okay, so we also have rate of mutation. Rate of mutation is affected by how often a change in DNA occurs, whether or not the change is detected and or repaired, and how likely it is that the organism is exposed to mutagens. What are mutagens? Mutagens are things that um, up the rate of mutation. So stuff like radiation. That's why we don't like to do a ton of X-rays, or why Marie Curie eventually died of the radiation she was exposed to, because it damages our DNA. So that's like a mutagen. Um, so let's dive deeper into the cause of mutations. So we have spontaneous mutations. That would be just a replication error or perhaps a wobble pairing, like an A pairs with a C instead of an A pairs with a T. Um, so that would just spontaneously happen, just an error blip in the universe kind of thing. We also have strand slippage. So when they're replicating, wrong thing. When they're replicating, you could accidentally have this strand like slip and maybe curl back onto itself, or this one bunches up and this one keeps going. So that's when usually a bigger mutation would occur. We also have unequal crossing over, which leads to indels. We have depurination, which is this you should know. It is an important thing. Cleavage of A or G from the DNA, and it is usually replaced by A. So basically, depurination, it's taking the purines out of the DNA. So if we have a sequence that goes A, G, C, T, A, G, depurination would be take out this G is cleaved out. So usually, because these happen so frequently, they just replace it with an A. So now it would be A, C, T, A, G. So that's how that goes. Of course, if this A gets cleaved out and it's A, G, C, T, A, then when it would be replaced with another A, nothing happens. So that's kind of a hit or miss with depurination. Deanimation is cytosine to uracil, but in general, what you need to know about deanimation is it usually makes the C to G replace with A to T. So it completely replaces that base pair. Other things are chemical agents. We also have radiation, as I already said, that causes oxidation, that causes free radicals, that breaks our DNA. We also have more radiation. Importantly, X-rays and gamma rays break the DNA, but not UVA. We don't like UVA. UV creates dimers in the DNA, which kill bacteria. So basically, a good way to sterilize something is to shine UV light on it, because it creates these dimers where basically the T pairs to the T in the strand. So if it was A, C, A, and this was T, G, T, A, T, these T's would pair together instead of pairing that way. So we would have pair, 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 and then these would not have a pair, and these T's would pair together, and then these A's would kind of be left out, and then pair, pair. So that's what a dimer is, and with a dimer, bacteria cannot replicate their DNA when there's dimers, so they cannot reproduce, and so they essentially just die off. So that's what UV does. DNA repair mechanisms. There's mismatch repair. Basically, uh, there might be kinking in the DNA, and then enzymes, because basically when there's a mismatch, say C and T together, which should not happen, this causes kinking in the DNA, and then enzymes help fix this. So they go, oh, no, that's not right. We'll do A and T. There's also direct repair, which they just convert the changed nucleotide to the original. It's kind of the same as this. There's base excision repair. So basically, they go, oh, this nucleotide you are leaving, and then a polymerase will come and fill that up, and then ligase will seal it up. There's also nucleotide excision. Um, the damaged strands are separated. They basically take out a huge chunk, and then normal replication machinery fills in. So this is like for a huge mutation. Like if we have normal and then we have like a mess here and normal, they will just cut out this chunk, take it away, and let replication fill that back in. There's also double strand breaks, um, which leads to homologous recombination, and it's kind of error prone. It's not great, but we already talked about that in the last unit. We also have translesion DNA polymerase, and this is kind of a coping mechanism because if there's like breaks or lesions in the DNA because the bases are mismatched, it knows to, in replication, that it can bypass those and it doesn't affect it as badly as, say, the dimers in DNA, which kill bacteria. That DNA polymerase cannot cope with that, whereas translesion DNA polymerase can cope with these dimers and it just skips over them and keeps going. We also have transposable elements, and these are genes that basically jump from place to place. Actually, a large percent of our DNA, 45%. And we have DNA transposons, which basically the DNA moves. And there's our retrotransposons, which the DNA turns into RNA, which is reverse transcribed back into DNA in a different spot. And sometimes it's copy paste, or sometimes they cut and paste. It just depends. And it relies on an enzyme called transposase. Um, and another fancy name for it is insertional mutagenesis. Uh, and sometimes they can cause silencing of other regions. I don't think you need to know that much about this, but do know that they exist. They are a thing, and they are pretty cool. Hello, and all right, so the next part, if you notice that we switched platforms, it was because I was trying something new. It didn't work out, so now we're back to our trusty OneNote. So yes, chromosomes and organelle data. Chromosomes can be one double-stranded DNA molecule and is circular in prokaryotes or linear in eukaryotes. It encodes thousands of genes, but there's also a large percentage is non-coding DNA. The majority of it is non-coding DNA. So um, regulatory DNA like promoters, or there could also be like telomeres, which are repeating sequences. I think we'll talk about that later if we haven't already. And they are very big. So we talked about plasmids before. They are small circular like pieces of DNA that have certain genes on them, but those only have maybe a handful of genes compared to what chromosomes have. They also do supercoiling. So if you've ever had a string and you start twisting it, um, and then all of a sudden, even after you start twisting it, it will start twisting up on itself. So then it will start coiling up really tightly, and you're not even doing the twisting anymore. It's just like the tension of twisting is making more twists happen. That's what supercoiling is. Ignore my bad drawings as usual. And this is helpful because it lowers the volume of space that the chromosome occupies, because these are huge and they need to be squunched really small so they can fit in a cell, and cell doesn't have to waste extra space and energy trying to keep the chromosomes all held together. They supercoil up so they take less space. This can also happen in circular DNA. So just because circle DNA is technically a circle doesn't mean it always stays a circle. It can also twist up and supercoil. So prokaryotic chromosomes, as we said, singular and circular. Yes, there's only one prokaryotic chromosome, whereas in eukaryotes there can be multiple. For example, in humans we have 23 pairs. They are contained in a region known as the nucleoid. So not the nucleus, but the nucleoid, and they might be anchored or organized around proteins. We don't really know. Topic of study. It kind of looks a little bit like if this is the cell, then we have it, the large circular chromosome, just like coming out in like little squiggles. So yeah, it's not really circular. We just call it that. And then we also have plasmids floating around. So these would be like little bubbles here and there that carry a handful of genes that are useful to the cell. And these can also be transferred between bacteria through bacterial transformation, and that's pretty cool. Bacteria cell division, we use theta replications for bacterial chromosomes. It divides in the middle, and then the plasmids are roughly equally divided. So if we have this cell here, then we would say, oh, you two plasmids are over here, this plasmid's over here. That looks about equal with the amount of plasmids we have. Okay, split. So now this one will have two plasmids, and this one will have one plasmid, as well as the chromosome. So that's kind of how that works. We have eukaryotic chromosomes, which are a teeny bit more complicated. So we have DNA, and then they're wound around these circular proteins called histones. And the DNA and histones combined make up something called chromatin. And they kind of form these structures where we have one protein, two protein, three protein, and they're all connected and wrapped around, and connected and wrapped around. And so that we call that bees on a string because it looks like bees on a string. Um, this main protein, they're referred to as histone proteins, but the main protein is also called a nucleosome. And these proteins help the DNA condense into more complex structures. Again, in tandem with the supercoiling, it helps them compact, stay small, which is good for the cell. Um, we also have a difference in chromosomes at different stages of the cell's life. So if we remember interphase, anaphase, metaphase, telophase, all of those phases of my mitotic, meiotic division, however you pronounce that word, um, we would have different types of condensing of the chromosomes because when the chromosomes are super condensed, they cannot be replicated easily. It's hard for DNA polymerase to get in there. It's also hard for them to be transcribed, so it's hard for RNA polymerase to get in there. So basically, when they are wrapped up, they're kind of dormant. Nothing's really happening with them, and they have to be loosened up for something to happen. We actually have special words for this. So interphase chromosomes are tightly wound, and those would be called heterochromatin. So these are tightly wound, they're not acetylated, and they're not expressed, they're not replicated. And then during mitosis, when the cell is trying to divide and replicate that chromosome, it gets loosely wound, and that is euchromatin. So that's acetylated, expressed, it's also replicated. So those are the two different. Um, those are two phases of chromosomes that you really need to know. We also have methylation. It suppresses gene expression. Demethylation activates genes. This is opposite from acetylation. A lot of people think, oh, acetylation and methylation, they kind of like if it's acetylated and methylated, then it expresses. No, they're actually opposite. So if it's acetylated, then it is expressed. If it's not acetylated, then it's not expressed. However, if it's methylation, usually it's not expressed, but demethylation actually activates it. Um, One way I thought of this when I was studying, which is a way that I thought was methylation. Oh, meth. Not great for your ability to do well on tests, meaning those like smart genes aren't being produced. So it suppresses gene expression. That's a really wonky way to go about it, but that's how it worked for my head. So I was like, okay. Um, we also have the centromere, which is basically we have the chromosome, and then we have well, this is all euchromatin. There's there's spot usually in the center, but it can be honestly in all different places. That's how we identify chromosomes. Um, in the center, that is always tightly condensed, and it kind of holds the whole chromosome together. And then we have a telomere. So these are repeat end of chromosomes to prevent degradation and sticking together, and they're usually just like repetitive, like 6 or 5 base pair sequences. We covered them in the previous unit more. We also have just in general, DNA has a bunch of unique sequences. It has even more repetitive DNA and highly repetitive DNA. And that's just a lot of that is like telomeres, where they're just repeats of the 5 base pairs. So a lot of DNA is actually like that, just really repetitive. Um, we also have a diploid, and that's a term we should know, and that means that you have 2 copies of every chromosome. So a diploid would be 2n. N is the number of chromosomes. N is the number of types of chromosomes you have. So in humans, that would be 23 because we have 23 types of chromosomes. And but we are diploid organisms, which means we have 2 sets of that 23, which brings us to 46. So that's why we're diploid. So 2n, so that would be 2 times 23 equals 46. So that's our human example. And these copies are homologous, which is another way of saying they're the same type of chromosome. They code for the same genes, they just maybe have different alleles on them. And different homologous pairs or different types of chromosomes are identified by the location of the centromere. So sometimes the centromere is in the middle, where you'd think it would be, but sometimes the centromere is actually up here, and it kind of looks like 2 little nibbins and really long tails. So we use where the centromere is to identify which type of chromosome it is, because that is fairly consistent for each type of chromosome. Um, in mitosis, we separate the sister chromatids, and then cytokinesis is separation of the cytoplasm. I know you in a different bio class, you probably had to memorize the phases of mitosis, but for this, you should probably maybe have a gist of them in the back of your mind, but it's not that important for this class. We have organelle chromosomes. So these are cool because mitochondria and chloroplasts were once they're individual organisms, and therefore they have their own chromosomes. So they have circular chromosomes because they were originally prokaryotic, and they have their own gene expression function. So they have their own ribosomes, they have their own RNA polymerase. However, they can rely on certain genes encoded from the nucleus. So the reason that we don't just have like bacteria living in our cells, mitochondria and chloroplasts are no longer bacteria, is because some of their essential functions rely on certain genes in the nucleus. So if they were just stranded on their own, they would die because, say, maybe their RNA polymerase is non-functional, and they use the RNA polymerase from the nucleus of the cell, and it goes all the way into the mitochondria, and then that helps it not replicate, translation and transcription, or transcribe genes. If this stopped giving it RNA polymerase, it wouldn't be able to express genes, and it would become essentially useless. And it wouldn't be able to carry out its function. It would die. So that's why these are no longer independent bacteria, but they still have their own replication methods. Inheritance for these are pretty interesting because mitochondria are passed down, both of these are passed down by one parent only. For mitochondria, it's your mother. So the maternal side passes down the mitochondria. You don't get anything from your father. So when we're tracing genetic trees, sometimes you can use mitochondria DNA to trace the mother's line of the family. Um, and also, interestingly enough, since the mitochondria is maternal, if there's a gene from the mitochondria, you know it was passed down by the mother. However, you can also have weird things where the mother is not affected by a mitochondrial disease, but the child is. So if the mother, say the mother is perfectly fine, but then the child's mitochondria is not working correctly, then you're like, well, how could this be? Clearly, the mother has to pass it down to the child. So did like a spontaneous mutation occur? Well, usually what happened is, do you remember how the mitochondria are dependent on certain genes from the nucleus? So perhaps the father gave out some genes into the normal 23 chromosomes of this person, and one of his genes is defective. But for another reason, or one reason or the other, he's fine, but he passed on a defective gene about the mitochondria here. So this gene, um, this mitochondria relies on this gene. So this one comes down here, and now this mitochondria is not functioning properly, not because of genes from the mom's side, but genes from the dad's side. Um, so that's kind of how certain weird hereditary patterns can happen. We also have something called heteroplasmy, which means that you could have different types of mitochondria in a cell. For example, if a mitochondria mutates, or perhaps different lines can get muddled, and whenever a cell divides, it kind of roughly splits its mitochondria, just like, or chloroplast, just like bacteria splits its plasmids. And sometimes you can get some where you get like, quote, a bad mitochondria and a good mitochondria. And depending on how much bad versus good there are, it will increase the severity of your disease. Um, and homoplasmy is when they're all the same. And segregated replication, that's what I just described here, where they're split off. And something cool is that when chloroplasts do this, sometimes if you've ever seen plants with like white stripes, white veins, or white patches or red patches, those variegations are due to the chloroplast splitting unevenly. And so they give off different colors, or only half of them are working correctly. So that's pretty cool, in my opinion. Diseases that can happen from for humans and mitochondria is usually a lack of oxidative phosphorylation. So if you remember from introductory bio, that this is a crucial step in producing the majority of ATP during cellular respiration, which means your body doesn't have energy, and that would lead to muscle weakness. And the severity can also relate to heteroplasmy, because if you only have a few defective mitochondria, maybe it's almost unnoticeable. Whether if you have all defective mitochondria or majority affected mitochondria, then it can be pretty bad. You can have fatigue, stuff like that. We also have the idea of horizontal gene transfer. This goes back to the idea that mitochondria and chloroplasts were once independent cells. And basically, how did these essential genes that they need to survive end up in the nucleus instead of where they belong in the mitochondria? So that's a question scientists are still trying to understand. All right, and then our next section we have chromosomal variations. Um, so first we need to understand chromosomal variations, we need to look at meiosis, which is not to be confused with mitosis. So meiosis is the process of producing sex cells that will eventually hopefully become offspring. Whereas mitosis is just a cell dividing. Um, like for instance, say your skin healing from a cut, then your cells has to divide to create new skin. Um, so basically it has two steps of division of the chromosomes. So first, it will line up all the homologous chromosomes just like in mitosis. This time it will separate these. So each gamete gets a chromosome. Then, during separation division, it will separate this cis chromosome into two sister chromatids. So, let's color these. So if this was green and this was red, it will make orange. It will make an orange one and green one. Um, in each gamete. So it would be in two separate cells. And the end result is 4 haploid gametes. If we remember what haploid means, that means it only has one set of chromosomes, which means in humans, this gamete would only have 23 chromosomes instead of our normal 46. Um, and errors during homologous pairing can cause mutations in the cell. And some kinds of errors in this homologous pairing we can see in a karyotype, which and these errors in pairing are called non-disjunction. It's when you have the chromosomes, maybe instead of only one being pulled this way, maybe two are pulled this way. And then you get a gamete that has an extra chromosome. And that's what happens in Down syndrome or primary Down syndrome. They have three copies of the chromosome 21 instead of just two. And that causes a whole host of errors because something called gene dosage, which we will touch on later. Um, so crossing over happens during meiosis, and that creates genetic diversity because basically if this homologous, these two chromosomes are together, it will be like, hey, I'll give you, if this one's red and this one's green. I don't know why I keep saying red. It's orange. I will say, I'll give you this and you'll give me this. So then we end up with a chromosome that looks like this. So it has um one part has a little bit from the green chromosome, one part of the green chromosome has a little bit from the orange chromosome. So that creates genetic diversity. We also have segregation of homologous chromosomes. That just means that they will all that it's not random chromosomes that are crossing over that would cause a whole host of problems. These chromosomes carry the same genes, they just maybe carry different alleles. We also have independent assortment, which means say the orange ones are from your father and the um green ones are from the mother. And so these ones, they won't always line up where all the father's chromosomes are on one side and all of the mother's chromosomes are on the other side. No, it's going to be something more like it's completely random. So it could be, oh, green ones on this side, green, green, and orange like this. And that also creates genetic diversity. There are few types of chromosomal variation. We have rearrangements, which affect structure and location of the chromosome. We have aneuploidy, which means you gain or lose a few chromosomes, like we see here in trisomy 21 or Down syndrome. We have polyploidy, which is the gain of a complete set of chromosomes, increasing n. So if you were 2n and you had polyploidy, then that would probably go to like a 3n or a 4n. So that means you have multiple pairs, multiple copies of the chromosome, more than you're supposed to have. And karyotyping chromosomes, which is right here, this is a karyotype. You can see the mutations. You can see you can see whether it's whether this person is biologically female or biologically male. Because females have XX and males have XY. So the absence of the Y means that this is a biological female. And then, so karyotyping, pretty cool. You can also see if there's trisomies or monosomies, meaning you're either missing a chromosome or have an extra chromosome. So that can look for huge chromosomal variation. Um, so now we have the huge topic of chromosomal rearrangements. So we have duplications, which have a few different types, but this is essentially part of the chromosome is duplicated. Not the whole chromosome, because that would be aneuploidy, but just segments. So we have first tandem duplication, and this means that the repeated segment is placed right after the original segment. So if you see here, we have AB, the repeated segment is AB, and it's just stacked right on top, and then it just continue, and then the chromosome continues like nothing happened. So that would be tandem duplication. We have displaced duplication, which means the repeated segment and the original segment have unduplicated segments between them. So if we have AB and the repeated segment is again AB, we have AB, CD, and then we have AB again inserted farther farther from the original than in tandem duplication. We also have reverse duplication where the repeated segment is flipped and just attached to the segment. So we have a little bit of a palindrome. So in this one we have AB, VA, and then continues as it goes on. And this can change the phenotype because of dosage effects. Now we say dosage a lot. What is dosage? Basically, if you have too many of the same gene for some reason or another, this is still something we're looking into. Um, it becomes too much for your cell. Too many options. Maybe the genes interfere with each other, but basically, if you don't have the right amount of dosage, which is two genes, two chromosomes, things start messing up in your cells and your body. So basically, if you increase the amount of genes, the same gene that you have, if you have normal humans would have two copies of the same gene, one from the mom, one from the dad. If you had, say, an extra chromosome, you had three copies of the same gene, that's overdosage, and overdose usually is bad, and in this case, it's also bad. Um, you could also have underdosage, which is also bad, sometimes a little less bad. It just depends on the gene. But when we're talking about dosage, that's what we mean. We have too much of one gene and it causes the cell to malfunction. We also have deletions, which is the removal of segments. And this is pretty bad because when we have duplications, it might affect the gene dosage, but we still have that core function that the gene codes for. But if we just delete a whole chunk, then it leads to loss of function. And loss of function is usually pretty lethal because, like, imagine if we were going into this test, but um, we didn't cover one whole lecture's worth of slides, and the information was just never given to you, but you were expected to know it anyways. That's kind of what the cell does. It kind of freaks out and and it is like, I don't have that protein to do that function. Oh well, and then everything falls apart. So that's why it's usually lethal. And also, if you lose the centromere, you lose the chromosome because that's kind of holding the chromosome together. So deletions are pretty bad. Um, what is the cause of duplication or deletion? Um, Often it's an unequal crossing over. So when the orange and green segments exchange segments, maybe the orange segment like holds on to a little more than the green segment, and so things get mismatched. Also chromosomes can break, or if they're broken and they're repaired incorrectly, then these things can happen. Um, we also have inversions. There are a few types of inversions. We have paracentric, which is an inversion that does not include the centromere, and pericentric, which is an inversion that does include the centromere. So, if we had a chromosome here, the centromere here, and let's just say for the purposes of this, that this side is green, no, this side is orange and this side is green. Pericentric, so one around the centromere would end up going orange. If the centromere is here, we'd go orange, orange, green, green. So if you can see, this little segment just got caught out of this one and flipped, and it includes the centromere, so it would be pericentric. Okay, so yeah, that would be a pericentric inversion, and then a paracentric one would just be that, but without the centromere. So we would just like invert a segment of the DNA, but that would not include the centromere, so it'd be either more towards top or more towards bottom. And this is caused by breaks in the DNA. It can have loss of function effects, positional effects. Um, that's inversions. You also have translocations. Sometimes a segment of the chromosome can move to another chromosome, like completely unrelated, doesn't even carry the same genes, um, different type of chromosome, or it can just have a segment of the gene can be moved to a different place on the same chromosome. Um, if it is translocated to a non-homologous chromosome, that's generally a lot worse because now we're on a completely different chromosome with different regulation, and it's just a mess. And this can be caused by double strand breaks. We also have different types. So we have reciprocal versus non-reciprocal. Reciprocal is the two chromosomes swap segments when they weren't supposed to. This is not crossing over. This is a bad thing. And we have non-reciprocal, which basically one of the chromosomes is like, hey, I don't like this one segment on myself. I'm going to cut this from my body and paste it onto another person because it's their problem now. So then they're just kind of like a gap in this chromosome. And obviously that's not very good. And now this chromosome has extra length. So that's pretty bad. That's what I mean when I say copy, cut, paste. We also have something called Robertson translocation, where we have two areocentric chromosomes, which means that they're very long, they have their centromere, and they have a short nib at the end, and then another very long and short nib at the end. Sometimes these can translocate, so then we create one super long chromosome and one little nib that is pretty much unusable. That's Robertson translocation. We also can talk about aneuploidy. So we touched on this before, but now we're going to expand it, which is the change in the number of individual chromosomes. These names are kind of self-explanatory. Null isomy, that means you're missing a set of chromosomes. So the calculation for that would be 2n minus 2. If you had a monosomy, mono means one. Then we are down one chromosome. One pair doesn't have its partner. How sad. We also have a trisomy, which is when we have three chromosomes instead of two chromosomes in a homologous, not pair, but threesome, I guess. And then we have a tetrasomy, which would have four chromosomes. So if say trisomy 21 was three chromosomes, tetrasomy would have four. This you don't really see in humans because basically like guaranteed death. We also have double trisomic, which would be 2n. So then that would have two homologous pairs that had an extra little buddy on them. So then you would still have an extra two chromosomes, but it wouldn't be all on the same type of chromosome. This caused non-destruction during meiosis. We talked about non-destruction earlier. Usually lethal. The ones that are least fatal are the ones you usually hear about, like Down syndrome. We also have Turner syndrome and Klinefelter syndrome. Those affect the sex chromosomes. And since the sex chromosomes don't have genes as critical to life, usually those have the highest survival rates. But in general, missing a chromosome or having an extra chromosome is pretty bad, and most um, of these pregnancies would result in miscarriages or death shortly after birth. Um, we also have mosaicism, which is when after you're born, you born healthy at some point during your life. Uh, one of your cells could have a weird happening during mitosis, and suddenly one of your cells has a trisomy, um, and one of them has a monosomy. Um, this could cause something bad, but also sometimes it can't. It doesn't, but it basically has variation within your own body. Um, Also, if say early in birth, this happens, then you could have yourself be like quote unquote 25% Down syndrome, which means 25% of your cells have trisomy 21 and the rest are healthy. So then you would have much more mild symptoms compared to someone with primary or all of their cells have triple chromosomes on their 21st chromosome. You have much milder symptoms than full. So that's kind of how mosaicism works. We have polyploidy, which means you obtain a whole extra copy, and these have a few terms you should know. We have triploid, 3n, tetraploid, 4n, octoploid, 8n. Those are kind of, if you know prefixes, those are pretty easy to remember. Uh, these are a lot more common in plants. They're really rare in animals because when it happens in animals, especially mammals, it's pretty much a death sentence as well. Um, but plants sometimes have like a ton of just copies of chromosomes. It's pretty cool. And these can be two different kinds. We have autopolyploid. So this has more than two sets of homologous chromosomes, but they're all from the same species or organism. However, these are rarely fertile because then say you have 3 chromosomes in meiosis because you're trying to reproduce, then like, oh, now we have to separate the chromosomes unequally, bunch of non-disjunction happening, so they're usually infertile. Which is why like you often hear, which is like a part of why stuff like ligers or hybrid animals are infertile, among with various other reasons. We also have allopolyploids, um, which is an organism with complete sets of chromosomes from 2 different species. So this would actually be more applicable to hybrid animals, but basically you have um, say if a lion and a tiger, we make a liger, then this organism now has sets of chromosomes from both a lion and a tiger, and in allopolyploids, they have more than just one copy from a tiger and one copy from a lion. Maybe they would have 2 copies from the tiger and 2 copies from the lion, which would make them an allopolyploid. And so this would result into a tetraploid, assuming they had 2N and 2N. And a tetraploid can be produced by various combinations of the parents, and I suggest for the test that you know this. Um, Also for your homework, assuming you are in my class. So now we have control of gene expression. This is our next topic. Gene expression is a regulated response to developmental cues, changes in environment, signals from other cells. So all of these are things that can impact gene expression. Um, Genes are often classified by function. Um, it's called gene ontology. We have various methods of regulation. So we have transcription regulation. This basically means we stop transcription or we promote transcription. And um, you need the first step in central dogma is to transcribe. So this is pretty crucial. And there's 2 types. We have positive, which means the regulatory elements are enhancers. So they say, yes, more transcription, do more of it. Or we have negative, which regulatory elements are inhibitors or repressors. So they either drastically lower levels of transcription or stop them altogether. And these proteins are produced separately from the gene itself. So, as we saw in the operon, which is slightly different from how eukaryotic cells work, but if you watched my previous video about the lac operon, you would see that these proteins are produced separately. So the LAC-I which is a repressor protein, has its own promoter, its own transcription, versus the rest of them, which has LAC-C and LAC-Y. If that does not make sense to you, I highly suggest watching other video because I think it will be helpful for you, especially on the test. We also have DNA binding proteins, which are basically these regulatory elements and they either promote or inhibit. We have operons, which are in bacteria, and they're a bunch of genes with relating functions that are clustered together. We have the operator, which is where the regulatory protein binds, so either the repressor or the enhancer. Um, we It also contains genes for the regulatory proteins right next to it, but with a separate promoter. So it's in the because it wouldn't make sense if the regulatory genes were regulated by the regulatory genes. That just wouldn't work. Nothing would happen. These can have 2 kinds of operons. They can be inducible and they can be repressible. So if they're inducible, they're usually off and they're activated by something. If they're repressible, they can be they're usually on, but they can be inactivated if needed. So you will often hear stuff like an operon is positive inducible. That means it's usually off, but then an activator will come and promote it and have it start transcribing. If it was negative inducible, that means that it would normally have a repressor bound to it, but then that repressor would be released when it would need to be activated. So, combine combining this and this terminology is key, and you probably know that. We have lac operon. Again, I go into a lot more detail on my previous video, but since I've already made one, I'm not going to go into detail here, but it is negative inducible. And another thing that I touched on my other video is F plasmids. So it allows for partial diploids of certain genes, meaning you have two copies that are slightly different, but they're code for the same gene. Um, it can also use a form of positive control. Uh, this is not super um important for now, but if you would like to read this, you may. It's an example of kind of positive regulation. We also have the trip operon, which is negative repressible. So that means it's usually on. But then it produces tryptophan, and when there's enough tryptophan, then it will activate a repressor, which will then bind and stop transcribing until all the tryptophan is used, and it needs to transcribe again. We also have attenuation, which modifies the RNA while the transcription is happening and usually stops it prematurely by creating these little loops, like hairpin loops, that like tangle up on itself and like make it impossible to keep going or translate it into a protein. RNA molecules can also inhibit translation because if you have strands of mRNA waiting for a ribosome, what if a complementary strand of RNA just like slap back on top of that? Then the ribosome wouldn't be able to bind, it wouldn't be able to translate. So that's another way it can be regulated. We also have chromatin remodeling, and we can kind of change the arrangements of those proteins the DNA is wrapped around. And that either allows or prevents RNA polymerase and transcription factors from binding to the DNA and transcribing. We also have histone modification, acetylation, deacetylation, methylation. I talked about this earlier. And sometimes the state of chromatin, hetero or euchromatin, can determine whether the organism is growing or not. Because if it's growing, it will need to replicate its entire genome, which means that the chromosomes need to be in euchromatin so that they can be replicated. And then also for eukaryotes, they sometimes have a regulatory promoter that controls the level of transcription, so it's not all or nothing. It's a little more nuanced, as most things with eukaryotes are, to be honest. But all right, so thank you for listening. Please let me know if you have any questions, and good luck studying.